Did it just click, Mike? Can you see it? Is it?
Yeah.
Oh, okay, good. Okay, so what we're going to talk about for the rest of the morning is measuring absorption, and what we're going to do for the afternoon is measure absorption. So we used to not do this lecture. We would sort of just wing it in lab, but it seems like then we'd end up giving the lecture later. So Emmanuel, I decided to be proactive this time, but I noticed that he's sitting at the back of the room. So you will chime in anytime. Chime in. Just stand up, or you can come to the front. Yeah, right. So quiet. Blushing flower. So how do we measure absorption? Well, there's a couple of different ways. The ways that we're going to show you in this class are how to collect a discrete sample from the ocean and bring it back to the lab and make some measurements with it. And so we'll talk about measuring samples in cuvettes, filtering them onto filter pads, and doing what we call the quantitative filter technique. We will also show you how to use in situ meters, like absorption meters, to measure absorption. I'll talk a little bit about integrating cavity absorption meters. I didn't bring mine with me because I didn't want to waste your time because it's just proving to not be what it is promised to be. And I can share those results with you if you're interested, but we have more important things to do with things that actually work. So I want you to remember the absorption theory that we talked about, because in your mind, every time you make an absorption measurement, you should go back to this and remind yourself of what is important about how to make these measurements. And it's important to know something about the incident light. It's important to be able to measure the transmitted light. It's important that you have a very short path length so that you don't have multiple processes occurring within that path length. It's important to either have a sample that has no scattering losses, or that you have captured all the scattered light, or that you can correct for the scattered light. But if you don't do any of those things, then you're not really measuring absorption. Okay? And so with each sensor, it's important to characterize how each one of those constraints is being handled in order to really convince yourself that you're measuring absorption correctly. All right. And again, contrasting this to when you're measuring beam C, where you don't want to measure the scattered light, where you want to just measure the transmitted light, it's a lot easier to make a transmissometer than to make an absorption meter, which is why we had transmissometers 30 years before we had absorption meters. Okay, so let's talk about the discrete spectrophotometry first. So this is what happens when you go out and you collect a water sample from your river or your coastal waters or your lake. And often what we do is we filter it. And filtering has two important characteristics about it. One, it separates your particles from your dissolved. So now you can actually begin to parse out absorption into different components. And so here is the dissolved, and here's the particulate that's suspended on the filter. The other thing is that often the particles are very dilute in the medium. And so if you measured a whole sample absorption in a spectrophotometer with a 10 centimeter path, you don't get a very strong signal. And so you would not have, you would have very large uncertainties in your measurement of the particulate matter in a cuvette. The AC meters have a 25 centimeter cuvette, and so it's a factor of 2.5 more signal that they're achieving. So, but even they are really challenged in some of the lower biomass regions of the ocean. So concentrating particulates onto a filter pad helps you a lot with signal to noise. Okay, so I was showing you these round cuvettes. Has anyone worked with these round cuvettes in a spectrophotometer? Yeah, they're a pain, right? You can never get the bubble out of this little corner right here. Yeah, and then, yeah, it's just a hassle. So we will be using square cuvettes. If you are going to be moving yourself into spectrophotometry, I highly recommend the square cuvettes because you just don't have the same hassle of measurement as the round ones. So we fill our samples with some dissolved fraction, and in the lab today we'll be doing the 0.2 micron filtered samples. There'll be an incident beam and a transmitted beam. And let's see, what did I say? So the question is, if we have a cuvette that looks like this, what's the biggest difference you can immediately tell between that and the theory? It's a thickness, right? It's a varying path length. So we will actually be measuring the absorption as a function of path length. I have 0.5 centimeter cuvettes, 1 centimeter cuvettes, 2 centimeter cuvettes, 5 centimeter cuvettes, 10 centimeter cuvettes. The AC meters are 25 centimeter cuvettes. So you have this range of path length, but none of them are infinitesimally small. And so what we have to do is make sure that the optical thickness of the sample is dilute enough so that we don't have multiple, well, if we had scatters and that there's no multiple scattering in there, if there's too much CDOM in there, the signal's too thick, then you're not in an optical regime where if you made a dilution series, you would get a linear relationship with absorption. You know, if you made a standard curve. If it's too thick, it starts to tail off. Again, almost like self-shading, the CDOM over here never sees the light, but it's contributing to the path length. So, not if you are at the point where you're limiting, where the amount of light is just not making it through the sample. Right, just like flattening of the absorption peak for phytoplankton, you can get flattening of the CDOM. And what's interesting, and you will see a lot of examples of this in the literature, if the CDOM concentration is too high, instead of getting a nice exponential like this, you'll get a flatter exponential. And what does that mean about your slope? The slope will be lower. And so you'll see a lot of papers published that relate the slope to the magnitude of absorption, and they say, oh, we've got this great relationship. We have really high, every time we have really high absorption, we have really flat slope. It must be completely different composition of material. It isn't. It's just that they had too long of a path length for their concentration, and so their slope got flatter as their CDOM went up. So we're going to actually do this exercise today in the lab so you can verify that for yourself. So, yeah.
It's a little bit off topic from that, but since you mentioned this, though, CDOM is very photosensitive.
Yes.
It degrades really fast with light. And that also changes not only the magnitude, but also the slope. Right?
Yes, it does. Yeah, it often makes the slope steeper, and it reduces the magnitude. Yeah, so when you filter, you want to do it in a dark room. My lab basically never has the lights on. My technician walks around by feel in my lab. Right? So and also when you scan, we set our spectrophotometer to scan from red to blue so that you're increasing the energy of the light as you're measuring it. If you started at this end, you could be bleaching your sample as you're measuring it. And so then you would get a steeper slope because it's being bleached and lowered as you move to the lower energy red wavelengths. So some of the other assumptions of this method, well, so one is the path length. What else are we assuming here? What?
Mixed.
We have to make sure that it's mixed, absolutely. We're trying to measure absorption, so what else do we need to make sure of? No scattering, right? So we filter the samples, and presumably you filter through 0.2, everything is very small. But, oh gosh, you just couldn't measure your sample today, so you froze it or put it in the fridge for a while. You might be forming some particles again. So you have to be really careful about how long you store your sample and how you store your sample. And it turns out that seawater samples that have been filtered store pretty well frozen. Freshwater samples do not store well frozen at all. So you're better off, if you can't measure immediately, if you're doing freshwater samples, store them in the refrigerator. Seawater samples you can freeze. But always freeze them in like amber glass, non-reactive glass. Don't fill it all the way up to the top, because of course as you freeze it, it expands and cracks the bottle. So give yourself some space. Yes, Kurt?
Why is it that saltwater preserves well and fresh doesn't? Is it like the salt preservative?
I honestly don't know, but I did the experiment after a pretty harsh review of a paper. And so we did the full suite of fresh to salt, and we froze them and melted them immediately, froze them, and sort of did time series of keeping them cold and dark versus freezing them and thawing them. And the freshwater samples, you just could never replicate what they were freshly filtered if you had frozen them. And
Maybe because, you get humic versus fulvic acids and one preserves well and one doesn't.
I don't know. I'm not sure if it was so much that, or if it has to do with the salt sort of interrupting the lattice structure of the ice. I honestly don't know, but someone here could find out. Yeah. Yeah, project.
I heard that when you freeze, you sometimes when you melt it back, you may form like colloids and stuff that might change all that, like backscatter.
Yes. So what I do is I measure it, right? So I melt it, I measure it, and then I filter it and I measure it again just to make sure. And it turns out that often you may be making some colloids, but the melted sample is closest to what it was originally. If you refilter it, it's lower. So the colloids, even though they have formed, contribute very similarly to absorption as they were before they were colloids. But you should do it for your samples to make sure that that's the case. The best thing to do is just to measure them fresh if you can. Okay, so what are the other assumptions? Did we say scattering? We assume that there's no scattering, right? Okay. All right. And so this assumption might fail if you have too much CDOM or too long of a path length. The assumption might fail if you have particles that scatter light. It might fail if the scattering is not collected, whatever scattering is occurring. All right, so another approach to, instead of measuring it through a cuvette of this configuration, is measuring it in an integrating sphere, an integrating cavity absorption meter. And I was just recently on a cruise in February with Rudy Rutgers, who's, where's he? Is he in Kiel? Right. Okay. Yeah. So he designed an integrating. Hamburg. Yeah. He designed an integrating cavity that's made of Teflon, which is a highly scattering surface. You fill the cavity with your sample, your CDOM sample. Then he takes it and he slams it on the counter to get all the bubbles out, and you're like, ah! But it's definitely made to be at sea. And it has a port by which light enters. The light is scattered to create a diffuse environment. And then you measure a small portion of that light, and it essentially relates, the amount of light that you're measuring here is proportional to the spherical amount of light in there. And so you can measure the absorption coefficient. And if there's any scattering, you collect it, because you collect all the light. So it's really a great approach. So try to filter so you don't have scattering, or try to get some sort of an integrating cavity absorption meter that works. Okay, yes?
Do they compare like both methods?
Yes. Yes, so we were on this cruise. It was at a closure experiment. So we collected water samples. We filtered them in triplicate. I got 3 samples. For each water sample, I got 3 subsamples. He got 3 subsamples. He measured his integrating cavity. He measured it with a waveguide, and I'm measuring it in a spectrophotometer. And so, yes, he's done comparisons, and now we're doing comparisons between labs as well. They look pretty close. He has a number of corrections that he goes through, which seem to, the end result seems to get to closure with our measurements. So, yeah.
Are we going to be measuring absorption or absorbance?
We'll be measuring absorbance,
So
the capital A.
what do you mean?
It doesn't have units. We measure the ratio of the transmitted over the incident in log 10 space. And then you will convert it to absorption through a conversion to natural log and dividing by the path length. So you need to know the path length of your cuvette to derive the absorption coefficient in units of per meter. Yes, because it's confusing.
And the confusion is even worse because the nomenclature is different between different fields. I'm dealing now with an editor in an optics journal that claims that absorption is at no use.
Right.
He's an adversary guy. He might be an adversary.
They say extinction. Anyway, they use extinction. They don't even use absorption.
So you just have to be aware that, like any cultural misunderstanding, different cultures have different, they use the same word to mean different things. I mean, pronotionality closure in physics, pronotionality is very different than closure in optics. Same word, very different meaning. The pronotionality has to do with how you distribute it, how you close the interaction, the turbulence effect on the fluid in the bottom of the fluid.
There's also absorbance.
It's probably the one log E.
That could be the log E of I over I naught. Yeah, I try to not absorbance. Oh, no, absorption. I'm writing what I'm saying.
And even the letters we use are different.
So the absorption coefficient in units of per meter is equal to the log base 10 of E, or log base E of 10. Log base E of 10 times the absorbance, which you measure, which comes out of a spectrophotometer, divided by the path length in meters. The path length is usually in centimeters, so just make sure you convert it to meters. Yeah.
How do you know which base to use if you're loading it?
So all spectrophotometers come out with this. So whatever a spectrophotometer provides you with, although read the manual just in case, that's what goes in here. And so we're calling it absorbance because we are saying that we're excluding scattering. But really what it's measuring is optical density. There could be scattering in there could be absorption in there. So spectrophotometers provide optical density. If there's no scattering, you can call it absorbance. So I hopefully made all the changes in all of the lab handouts to absorbance. But if I didn't and I have optical density in there, just know that the optical density is what you're measuring absorbance if you're assuming that there's no scattering. Okay. So the other end of this is, so that's CDUM. Short and sweet, because there's no scattering. Yeah.
So how many people We have a few students who are already done it. If you get a curve of CDOM that does not go to zero in the near infrared, what do people typically do? Somebody said here subtract out the near infrared. Have you done it? Have you seen it done?
I've never set to zero in the 800 or something.
You said if you force it to be zero,
Yeah.
it's 100.
I don't remember some sure.
Everybody can think why you should or not do that. Has it ever been justified?
They're all set to be like backscattering and all that.
Of what? No, you're totally
You-
right.
you-
I mean, that's the conventional wisdom is that when you do basic scattering, if you see CDOM is not equal to scatter, you're going to remove it together. But, and again, as brother said, remember that point, because we'll come back to it when we talk about scattering. Because depending on what it is that's scattered, removing a flat line might or not.
Be the right thing to do.
be the right.
So another thing to think about is if we just look at this, say from the green to the yellow to the red, and it's a constant low value, it's really hard to think about the type of molecule structure that would lead to a constant absorption over a broad wavelength range. There's often a peak and tails. And so, so part of the justification might be, well, it can't be absorption because the physics of absorption don't predict this type of behavior. But then it means that you've got scattering in your cuvette, so then you need to think about that. But you have to think really hard, because if we had this same sample, but maybe it was twice as thick, or maybe we doubled our path length, we would have twice the absorption. So now it looks like that. And what if it was even thicker? And you get to this point where, if it looks like this, like that's a lot of absorption to have to remove. So just because it's really small, you're like, oh, I'm just going to subtract it. You have to, in your mind, think like, would I subtract it if it was multiplied by 10? This is an active area of research. There's some recommendations of what to do, but there's still uncertainty in these measurements.
And whenever you're studying these things, and just because people tell you subtract, think about why.
Yeah.
Why am I subtracting? Is there evidence that I use it? Always put to question everything that has. Is there a paper that explains why we subtract?
And what I'd like you to think about also is, if this is scattering, what size particles are in your cuvette? Tiny. Okay, tiny particles. And tomorrow we'll talk about scattering. And we'll talk about the spectral dependence of scattering as a function of particle size. Okay, so we're not going to answer this now, but think about what spectrum of scattering you might want to remove. Is it flat? Is there some dependence? So keep this in mind. Okay, so today in the lab, we're going to measure CDOM a bunch of different ways with a bunch of different size cuvettes, a couple different natural samples, some really high, some not so high, one saltwater, one freshwater. We'll test all of these, this theory. Okay, the next day, we're going to go at the other end and look at what's happening to the particles. Okay, so first, we're going to talk about this quantitative filter technique. And this is important because now we can look at the absorption by the particles in the absence of CDOM, and then we can also do some clever tricks with that. So, the state-of-the-art way to measure the absorption coefficients of particulate matter suspended or filtered onto filter pads is in an integrating sphere. And it's in the type of integrating sphere where you can place the sample inside the sphere, not on the outside, but inside the sphere. And we have one that we'll that's set up in the lab, and we'll show you how to use it. The reason, of course, is the same that as you have your incident light coming in and you measure the transmission of light through your filter. But these filters are made of glass fibers. They're white. They're highly scattering themselves. And so if you don't collect the scattered light, you're really overestimating the absorption coefficient. By putting it inside the sphere, it collects all of that scattered light. Okay. How many people have spectrophotometers in their lab and they've measured particulate? Have you measured particulate absorption on a filter pad? Has anyone measured particulate absorption on the filter pad? Just one? Two? Do you have an integrating sphere? Good. Do you have an integrating sphere? Good. That's great. Most people don't because they're expensive and, you just don't have them. A CBAS, the NASA data repository, is filled with data collected not in an integrating sphere. So should we throw it all out? No. We need to find a way to correct it so that we can have the most confidence and the least uncertainty, and that moving forward for labs that don't have integrating spheres, we can still believe the data as long as we know what the uncertainties are. So we'll talk about that. But this is the state of the art. If this is how you want to make your career, you should buy an integrating sphere. It's the best way to go. So, yeah.
How do you, stupid question, just the first time, how do you deal with photons that go back into the sample and get scattered back into the sample?
Yeah, exactly. So, okay, so the first thing you want to do, anytime you make a measurement, I forgot to mention this, sorry. Anytime you're making a spectrophotometric measurement, you have to have reference material. So if I went back to the CDOM and I measured this, what is my reference material? I have to make a measurement of the cuvette with something in it as a baseline. Salt water or pure water? DI water?
We usually, for salt water, as far as I remember, we used to do artificial salt water.
Artificial salt water?
So if we have 35 saline, we need to salt water.
Okay, so because what you're interested in is the absorption by the organic material, right? So in the visible, salts don't make a big impact on the absorption. They make a huge impact in the UV, down around 200. If you filter seawater as a blank, is that what you were suggesting? It's the same as your sample, right? So you want to either make artificial seawater, but you have to be very careful that you've treated the artificial salts and combusted them and removed all impurities. Right? I personally use milli-Q water, pure water, as a reference. Your choice of reference is important, and just make sure that you can justify it. So what we'll show you today is we'll do some measurements with milli-Q water, with reverse osmosis water, and with tap water. And you probably will never drink your tap water again. Yeah. Anyway, so similarly, when we're making measurements of the particles on the filter pad, we need to have a baseline scan. And so you use a blank filter that has been wetted, that is wetted to the same degree that your sample will be wetted. So if you're filtering a liter of sample through your fiber filter, you'll want to filter about a liter of water, of pure water through it, because then it gets flattened and it changes the shape of the filter. So you want to treat your baseline filter the same way you treat your sample filter with whatever your reference material is. I use milli-Q water again, okay? Or you could use doubly filtered seawater. So you're going to make a bunch of measurements with blank filter pads, and those you're going to subtract from your measurement. And then you take your sample scans, and the experiment that I did with Rudiger and Daria Stramsky in January, we each got 3 filters from the same sample. We each measured each filter 3 times at 3 different rotations, 3 times amongst 3 labs. And I'll show you. It turns out once you do that, you can establish for yourself that the difference in rotation and the difference between filters, if you're a good filterer and you're really good at keeping your sample well mixed, are just, you can't even measure the uncertainty. So that's what you're aiming for. But it's good to establish that. Then you're going to subtract your baseline from your sample scans, and then you'll compute the absorption. And just like I said, this is the conversion between log base 10 and log base E, optical density over the path length. And so now the question is, what's your path length? In the cuvette, it's easy. It's the geometric length of the cuvette.
Is there filter volume?
There is a filter volume. And hopefully you've written it down. So do I just divide by the volume? I take a water sample. Take a water sample. I measure it. I have a liter. I filter it through a filter. And what's my path length? Well, if my filter was as big as this bottle, right, then this would be my path length. Because it's the same as measuring light in, light out, right? But if my filter is this big, I have to take this volume, squeeze it to this big, and stretch it out, right? So it's essentially like taking a volume of water and creating an area of your effective filter diameter, which would be whatever that diameter is here. And so the volume is the area of the filter, the effective area, and you should measure this with calipers, times the height, right? So the volume that you filtered, a liter, is equal to the effective area, which you've measured, and you solve for the height. So if it's a little volume will make a little height. A big volume will make a huge height. And so that's why we often filter particles onto a filter, because it's essentially increasing our path length, a very big path length in it for a dilute medium, okay, which gives us a very strong signal to noise. So the length, because we're having light coming in here and we're measuring the transmission out, it's the same as light coming in here and measuring the transmission out, as if it were in solution, but without all the water and CDOM. And so the length, the path length, the optical or geometric path length, is the height of this cylinder. And so that's the volume that you filtered divided by the effective area of the filter. This is not absorbance. So I don't like using absorbance, because there's too many A's. This is the effective area in units of meters squared of your filter. So pi r squared. Is everybody with me on that? I see some furrowed brow. I see a furrowed brow.
I'm not exactly sure. So this picture is like above your...
So this is an integrating sphere with a filter pad in it in cross section. So I took this filter and I placed it inside my integrating sphere, and I have an incident light, and I'm measuring some fraction of the light that's in that sphere that has gone through the particles and been attenuated. To figure out...
Where do you measure the light?
Down here.
Okay.
Yeah. And so if you take a volume of water and you put it through a filter of this area, it's as if you take your sample, you squeeze it to the effective area, but then that lengthens it up. And so you could sort of unpack the particles into a volume of water of a certain height, and that would be the path length over which you're measuring. So, let's see. There's my filter, there's my volume of water, and I haven't filtered it yet. It has some phytoplankton in it. Coccolithophores, I guess, because they're chalky. And then after I filter it, those particles are now highly concentrated in an area. The absorption of this, if I ignore the filter, is the same as the absorption of this, as long as I haven't packed this up too thick that it's like multiple layers. So these are equivalent. But the path length is not this. It was this when I collected my water sample, because this I took out of the ocean. And so that would be the absorption that I would measure in the ocean. Does that help? That's the path length. So, yes, when you collect your sample, you need to write down your volume, convert it to cubic meters. You need to measure your effective area. I measure it for my particular filter rig, and we've done a series of measurements so I know which filter cup results in which area. I keep track. So, because it's variable, and you can figure out when you do the calculation, if you have a little bit of error in your radius, it converts, right? It shows up right here. So you can actually propagate your uncertainty there. Okay. So what about scattering by the filter? So the filter itself scatters, and it scatters through the particles, the light, and the integrating sphere itself scatters. And so we have to account for that because it's an increased opportunity for reabsorption. So what do? Well, if you make this measurement, it looks like this. You can actually see the light scattering off of your filter. It creates a nearly isotropic light field. I mean, it's nearly equally scattering in all directions. And if you actually do some theoretical modeling, you can figure out the average number of number of times that the light will scatter through your filter. And it turns out it's like 2. And so you're essentially overestimating absorption by a factor of 2. Okay. But you want something a little bit more satisfying than that. So there are a couple of ways to correct for this. So what Dariusz Stramski just recently published a paper where he took some cultures or some really, really thick samples, and he measured them in suspension in his integrating sphere in a cuvette, and he subtracted the filtered sample, so he was just looking at the absorption by the particles. Then he filtered that sample onto a filter and measured the absorption of the particles. And then he looked at the ratio between those just to see what it looks like. So the measurement on the filter is what we call the actual optical path length, because the photons are scattering all around. They have a big path length, not just through the particles. When you measure it in the suspension, you can actually calculate what the geometric path length is. And so what you find is it's about a factor of 2. So if he plotted it opposite of what I would have done, but this is the suspension and this is the filter. And you can see that when the suspension optical density was 0.1, the optical path length through the filter pad was like 0.4. So in this case, it was really high. So he came up with a relationship that said, if I measured the optical density on my filter, I can correct it for what it would have been had it been in suspension, and correct for the fact that you've got the scattering. And that's called the beta effect. And so I've given you the relationship here, and it's in your handout. And so that's how you correct for the filter pad. And then once you've corrected for the filter pad, then this goes back into this equation to calculate the absorption coefficient. Yeah?
Why did subtraction of the blank correct for that?
Because the blank is used to measure the instrument signal, and there's no, it gives you, well, interestingly, the blanks are pretty flat because filters scatter light but don't absorb it. And so when you actually measure in an integrating sphere, your blank is pretty close to 0 except for some, features of the lamp or the detector. if you measure a filter in a transmissometer mode where you're not in an integrating sphere, you get this huge signal because it's scattering so much light out of the detector. But what you can't do is predict how much of that light goes through your sample multiple times and essentially increases the path length of absorption, the optical path length. It corrects for other things. Yeah. Yep. Yeah. It has an instrument noise as well, but it's pretty low. So. Okay. So then you compute absorption. Okay. So we'll do that tomorrow. If you don't have an integrating sphere, you typically use a scenario where you have a reference beam that goes through place of interrogation. You have a sample that goes through, or a sample beam that goes through your sample. You can measure by a photomultiplier tube or a diode array, depending upon your spectrophotometer. But what you notice immediately is that you're losing all of the scattered light in this mode because the detector's over here. And even if you have a filter here and it's losing certain amounts of scattered light, they're not exactly equal because this has absorption on it too. And so it becomes pretty tricky. So usually what we do is we try to match the optical density of the filter pad with something over here. This is a wet filter with sample on it. You can use a wet filter, but you have to keep replacing it because as it dries out, it changes its optical properties. So I, in fact, just use a neutral density filter that's made of quartz that has the same optical thickness as a filter pad, but doesn't vary in time. And then I take a measurement with just a blank filter pad as my baseline, and then I can measure my sample. And so if that's a setup that you have in your lab, I'm happy to sort of walk through that a little bit more. But the biggest problem with this is that you're not collecting the backward scattered light. And you still have to do a beta correction. And so Darius also calculated a beta correction if you measured your suspension in the integrating sphere, which is truth, and you measure your filter pad in transmission mode, which is not truth. And you can look at the ratio of the optical to the geometric path length, and here this is about a factor of 2 in this scenario. So again, you would do this correction factor for your filter absorbance, and then this is the number that goes in right here. Mm-hmm.
Just so I can understand, the upper one is water, right?
It's water with your It's a cuvette with your whole sample in it.
You take out the water absorption out of it to do this?
Yes. So you would first measure it, your whole sample, in a cuvette. Then you would filter it, and you would measure the CDOM essentially, and subtract that. So you would just get the absorption by the particles, because this is just particles.
So can you explain again why you have this factor of 4?
In the transmission mode, it's just a factor of 2, but the reason is because the filter pad itself, first of all, you're losing half of the scattering from your detector. And the filter itself is highly scattering in the interior, and so photons don't just go through and either get absorbed or get backscattered. They get scattered around in here, which means that they have a greater opportunity for absorption. So instead of...
What happens in the water? No?
Yes, but it's a very short path length, and so they only scatter once. Whereas here, instead of having a photon go through and either get absorbed or get scattered, you might get a photon that goes over here and then gets absorbed, or maybe scatters around inside the filter here and gets absorbed. So although the geometric path length can be calculated from the volume, the optical path length is this plus this plus this plus this. And you don't know what that is. Because when you measure that, you control how optically thick your sample is by either changing the thickness, the width of the cuvette, or the dilution, so that light comes in and maybe gets absorbed by that particle and it doesn't come out. Or maybe it comes in and it gets scattered, but the geometric path length is virtually the same as if it came through like this. You make sure that you don't have a lot of particles in there, because what you don't want to have happen is that. And so we maintain the optical thickness of the sample to be within a very small range, so that we can say that we don't have multiple scattering events in the suspension. But on a filter pad, there are multiple scattering events, and that's what you're correcting for. Yeah?
What about the light being scale effect from the integrated sphere onto the
That's what gets correct. Oh, you mean in here?
Yeah, because they're using the same.
Bless you.
Yeah.
So most of the light, by the time when it gets to here, because it's created this diffuse environment, most of the light that gets detected here has been scattered just one time through the cuvette. And what's really interesting, and it gets back to this, if you put a culture in here, I just did this on Friday, you put a culture in your integrating sphere, and you measure its absorption in the integrating sphere. It looks like this. There is no offset. So the particles are scattering. They're phytoplankton. They scatter light. But all the scattered light is corrected. And so you get no signal in the infrared. If you have a sample that has a lot of detrital matter in it, or a lot of other particles that aren't absorptive, a natural sample, you might get something that looks like you've got some phytoplankton. Looks like that. So phytoplankton don't absorb in the infrared, but non-algal particles appear to. Even if you keep them within the same optical thickness, you always see some near-infrared signal in the integrating sphere that you don't see with phytoplankton. So put that in your thinking cap. Okay. Emmanuel is a huge uncertainty guru. And will spend a lot of time talking about uncertainty. I am a convert. I've been a convert for a while about putting error bars on things. They're not pretty, mostly because most other people don't put error bars on things. So when you do, when you show your uncertainty, it's like, whoo, right? But if everybody starts putting error bars on all of their measurements, then everyone will, feel good about, well, I have error bars, but they're not that bad, right? But really, you can't distinguish between two different samples if you don't have error bars, right? So here's one way that we do it on filter pads. So here's your equation. So I run between 3 and 5 blank filter pads to give me a sense of the variability of the optical density of the filter pads. And then I compute the standard deviation of those scans. And I call it sigma for standard deviation. And then when I do this calculation for absorption, I also do it for my filter pads. So I put this standard deviation spectrum, which is the standard deviation of my filter pad measurements, I put it right into this equation, and I use the volume that was for my associated sample, because that's uncertainty is in my sample measurement. And then I calculate an absorption. So it turns out the volume is different for all your samples, so you have to do this calculation for every sample. And so it's just that equation with that spectrum in there. And so this is what it looks like. So here's an example. I have two different samples. I filtered the same volume, say 100 mils, but the optical density of sample 1 was 10 times as high as the optical density of sample 2. Just there was more stuff in it, and I filtered the same volume. I put that sigma spectrum from my blank filter pads into the equation with 100 mils in the volume. Those are my error bars. So here's the high particle water, here's the low particle water. I have a nice absorption spectrum for each, right? But look at the uncertainty. The uncertainty is due to the variability in the optical density of the blank filter pads. When you have a very low signal, when you have a very low absorbance by your sample, because it was just a low biomass water and you didn't filter very much, the uncertainty of your filter pad is large relative to your sample.
But is it the same magnitude as the high particle one?
Yes, it is. But it just, in comparison to your signal, is very large. So, same uncertainty, but because your samples had different optical densities, the relative uncertainty is larger. Okay? So, the other, yes. Is that a fair comparison? Yeah, it's a fair comparison. What it says, you should have filtered more. You had really low signal. But it's absolutely fair, and you absolutely would publish this. Hey, you might have filtered 4 liters. It could have been a sample where you filtered 4 liters, but you just don't have very much stuff in there. But the reality is, if your optical density of your sample is small, and the optical density variability of your filter pad is large, that's your uncertainty. So trying to compare this sample with some other adjacent sample when they both have a big uncertainty, can you tell me that they're different? I don't think so, not with this amount of variability, but you have to be honest about it. So the other is you filtered different volumes. Say I had the same scenario. I had very high particulate waters, I had very low particulate waters, but I'm going to filter a lot more volume in that low particle water. I'm going to filter a liter. Right? Now, when you go through the calculation, you get this. It's the same variability. The difference is that I took my low biomass waters, I filtered a ton more, I had a much bigger optical density signature, and so relative to the uncertainty, now it's small. So what the answer is, filter more volume. Don't measure samples where you have where it's really light and you have a really low signal, because you still have a very high uncertainty of your blank filter pads. Okay. This you would want to publish. Right? Yeah.
Doesn't the optical depth depend on wavelength?
Yes.
So wouldn't you expect the uncertainties, the relative uncertainties, which is in wavelength where you have very little absorption, to be larger?
No, because it's not the uncertainty of your sample, it's the uncertainty of your filter pad. This is just the filter pad. And it's pretty flat, pretty spectrally flat. Till you get a little bit out here, and then it's all over the place. Yeah, okay. So I would give you an example. Here's the optical density of a single filter pad rotated 3 ways. That is very good replication. Here are 3 different filters. That is very good filtering replication. Here's the uncertainty when you put all of those uncertainties and you calculate, the sum of the square error, and you do all of your propagation of error, and that's what you get. So this is what you're aiming for. Then you would put the filter pad of your blanks uncertainty on top of that, the uncertainty of your filter volume, and you would add all of those up, and that's how you should be presenting your absorption. Okay. Yeah. That it looks like what? I think the error bars are just, it's because you're on a very steep here. And I only did an error bar every 10 nanometers just because it was pretty, yeah. Okay. So, scan it. The next thing you could do, we talked about extracting with methanol. That gives you a non-alkyl particle spectrum. And then you can subtract the non-alkyl particle spectrum from the total spectrum and get the phytoplankton spectrum. Okay. That's why we do filter pads. Otherwise, they're just really a lot of hassle. But you do it because you can separate the phytoplankton from all of the other particulate matter. And we talked about doing organics. Here's the nice case where you muffled it and then you got to measure the absorption by the mineral particles, but it's like only 1 in 100 that work out, so I don't recommend that. Note that when you do this extraction, phycobilipigments are not methanol soluble, so you can gently treat them with almost boiling water or phosphate buffer, and that will extract the phycobilipigments out. If you notice that you have a nice detrital spectrum with some peaks in here, yeah, another treatment with water will extract them. They're water soluble. Okay, so we'll review this tomorrow in lab, but you'll have this handout. So thinking about how you're going to make a measurement. In all cases, your optical density measurement should be between 0.1 and 0.4. That's the golden, the sweet spot where you have enough signal to have a strong signal to noise, but you don't have so much material absorbing that you're out of the linear range. So those are numbers you should just keep in your hat. So if it's phytoplankton, that might be your red peak. Will be around 0.1, and your blue will be around 0.4 to keep yourself in this range of between 0.1 and 0.4 in optical density units. If you're beyond that, it starts to become nonlinear. So you don't want it to look muddy. Always think about what your reference is. The advantages to extract to separate non-alkyl particles, and then computation. Do you do an offset correction? Stramsky and Baban said that you should be able to do an offset correction. He now does not believe that. He now, from the integrating sphere work, does not do an offset correction in the integrating sphere. In a transmission mode, you don't know what it is. So this is a big question. And then you calculate your absorption from your optical density for particles, your optical density for non-alkyl particles, and then you calculate absorption of phytoplankton from the absorption. You don't calculate absorption by phytoplankton from the optical densities. It's not optical density of particles minus optical density of non-alkyl particles. You have to go to the absorption first and then calculate phytoplankton. Okay. All right. Phew. Everybody take a breath, man. There's just no oxygen in here. I'm gonna open up the door. Okay, so that's just in the lab. Now we're gonna move to the field. Or outside. How many people have worked with an AC meter before, a wet lab's AC meter? 10, 10, 2, 3, 4, 5, 6, 7, 8, 9. Okay, half. Excellent. Hopefully even you who have worked with them a lot will learn something today in lab. Okay, so these are quantitative measurements of absorption and attenuation. They have two paths. Has everybody seen an AC9? Is there anyone who hasn't seen one before? You haven't seen one? You haven't seen one? Okay, good. All right. So they're big. They have two tubes. They are designed to have a pump attached to them. And so water is pulled through the tubes and then pumped out. So the water goes through the instrument before it goes through the pump. And that's designed so that you don't break things. They still break things, but not as much as they could. One tube measures absorption and the other tube measures attenuation. Okay. C and A. If you subtract the two measurements, you get B.